Hello, today we'll talk about the fifth topic is the innate immunity. So innate immune response non-specifically combats invading microbes. Pathogens that penetrate the external barrier, for example, through a cut in the hand, encounter three types of non-specific innate immune responses. And these three types of non-specific innate immune responses include protection by white blood cells, either phagocytes or natural killer cells, or the inflammatory response and the fever. Phagocyte and natural killer cells destroy invading microbes. So phagocytes and natural killer cells, as we said, destroy invading microbes. Phagocytes do this job by phagocytosis, while and there are three types of phagocytes: the macrophage, the neutrophils, and dendritic cells. The neutrophils are active against bacteria, while macrophage against fungi, sometimes bacteria as well, while the dendritic cell active against any virus-infected cells. While for the natural killer cells, they are non-specific, although they are non-specific, but they attack only the majorly the virus-infected cells. The NK cells are not phagocytic. NK cells destroy virus-infected cells and cancer cells. They release proteins that make holes in the membrane, and through these holes of the that are made on the virus-infected cells, NK can secrete enzymes through these holes to kill the infected cells. So how do NK cells distinguish between virus-infected cells from the healthy cells? So normally the surface of our normal body cells displays a set of proteins, which is collectively called, of course, MHC, major histocompatibility complex. These MHC proteins are very unique to each person. They are only identical for twins. Twins have the same MHC proteins. The normal pattern and types of these MHC proteins are termed self. These are recognized as self, while virus-infected cells often lack some of these MHC proteins. So there is altered pattern of these and components of this protein, and they look non-self to NK cells. NK cells patrol the body, killing any cell that exhibit these non-self markers upon encounter. Most cancer cells have missing or altered MHC protein. Therefore, they can be identified by the NK cells as a non-self, and they are treated the same way as virus-infected cells. So you can see here from the figure, a self-marker can help the immune cells to distinguish that, while if a non-self appearing, in this case it's termed like an antigen, because antigen is a molecule that the immune system can recognize and respond, and it is usually non-self. And from this animated figure, you can see the process of phagocytosis. So you can see this phagocyte, and you can see it can phagocyte. Here is the here in this one. It is internalized now. You can see again. Here is internalization, the process of internalization inside the phagocyte. And as well, in this animated figure is a very good one. You can see how the NK cells can make pores on the cancer cells and secrete enzymes inside these cancer cells, and leading to its destruction. So actually, they managed to do this kind of experiment by labeling the enzymes that is inside the natural killer cells. And once in action, they can track these enzymes inside the NK cells. And it proves the action, and you can visualize the action actually of the natural killer cells. So for an innate immunity, it's a cellular response by leukocytes, white blood cells, that targets pathogens. That cellular response targets pathogen and usually decorated by chemical indicators like complement and the antibody, as we said in the last lectures. And these complement and antibody is derived from inflammation or even the adaptive immunity. So this innate immunity actually, as we said, provide the first line of defense from infection in non-specific manner. It is rapid and independent of antigen, which is obvious because it's non-specific. The effector cells, or the main players of the innate immunity, are granulocytes and polymorph nuclear leukocytes, PMN. Granulocytes actually, it's full with secretory vesicles of a lot of proteases and a lot of different enzymes, while the PMN have a very characteristic shape nuclei, and these kind of cells are attracted to the chemical indicators like the cytokines, they can deliver a cytolytic attack using chemical warfare. So what are the kinds of chemical warfare they can use, these PMN leukocytes that can use to deliver a cytolytic attack? Three types: free radicals of oxygen, like the oxygen free radical or hydroxyl free radical, antibacterial peptides like defensin, apoptosis-inducing peptides like granzymes. Okay. So here, when we are dealing with innate immunity, we'll talk about 4 subtopics. What is innate immune response? What are the cell adhesion molecules that facilitate the process of innate immunity? And as well, we'll talk about the pattern recognition receptor, PRR. And as well, we'll talk about the different types of the leukocytes. Before we talk about these subtopics, we need to understand that the innate immunity is divided into innate cellular and the innate humoral. Both of them are invariant rapid response. And what's meaning of invariant? They don't change. Okay. That's why it's a very ancient, and it is genetic imprint. This immunity is a kind of genetic imprint. While for the adaptive as well, it's divided into adaptive and humoral, and it's a variable which is different than the innate, and the response is slower than the innate. And a lot of factors, as we will know in next lectures, a lot of factors describe or make it, which actually is the reason why the adaptive immune system is very variable, like the somatic mutation, as well antigen presentation and the germinal centers. It's not the germinal system is not a factor of variability, but it's like centers where the adaptive immune response cells are concentrated. Natural killer cells and dendritic cells always play like connecting between both type of the immune response. For the innate cellular, you can see the main player beside the macrophage, the mast cells, the neutrophils, the xenophiles. For the innate humoral, you can see the complement proteins, the antimicrobial peptides, and as well the acute phase protein. For the adaptive cellular immune response, of course, the helper T cell, the cytotoxic T cells, and of course the B cells, which after that will form the antibodies. For the adaptive humoral branch, of course, the antibodies with the different types, G, M, and A and E. So, talking about the innate immune response, actually it's a sequence of events beginning with recognition of infection by either BAMPs or DAMPs. And these will stimulate the leukocytes as mast cell and macrophage. The chemokines, which are kind of a cytokines, the chemokines, of course, recruit additional leukocytes. These cytokines provoke specific physiological responses. Cross factors like colony-stimulating factors induce hematopoiesis, of course, which is obvious to increase the amount of cells in the blood. Bone marrow as well restore the circulating leukocytes. Of course, you know that leukocytes will extravasate into the site of inflammation and as well can park in special niches such as the lymph node. And of course, the main function of the lymph node is a kind of like filtration of the blood of any antigen born in the blood. Leukocytes are generally short-lived and die by apoptosis as the inflammation resolved. Okay, let's see in this figure the sequence of events of the innate immune response. So initially, tissue damage, as you can see from the figure, releases DAMPs to the surrounding area. This is followed by the pathogen can gain entry, either a bacteria or a worm can gain entry while presenting the PAMPs, specific molecules on its surface, the PAMPs. Soon after that, the complement and antibacterial peptide begin immediate response. So the immediate response is carried out by this molecule which is near the vicinity of the site of the start of the inflammation. Soon after that, the residual macrophage and mast cells are activated by these complement proteins, as well by PAMPs and DAMPs. So as we said, the resident macrophage and mast cells are activated by already the in-vicinity complement and antibacterial peptides. The macrophage start to secrete cytokines that recruit neutrophils and dendritic cells and more macrophage. So once activated, the macrophage start to secrete these cytokines, as you can see from the figure, the different cytokines that it can secrete: interleukin-1, CCL8, interleukin-6, TNF-alpha, prostaglandins, and leukotrienes. And as well, mast cell initiate the vasodilation or constriction and the relaxation of the tight junction. And obviously, it will relax the tight junction for the subsequent extravasation of the other leukocytes to facilitate leukocyte extravasation. These mast cells start to secrete histamine and as well prostaglandin and leukotrienes. Soon after that, the selectin the expression is increased of the selectin on the endothelial cells. This as well will facilitate the margination and the extravasation of the leukocytes or the white blood cells to the site of the inflammation. Soon after that, the neutrophils begins massive phagocytosis and cytolysis. You can see it starts as well doing phagocytosis and cytolysis. Soon after that, the macrophage as well activate the immature dendritic cells, as you can see from the figure, and leading to its recruitment to the site of infection. And these dendritic cells can scavenge the debris from the wound site, and the scavenging of this debris will lead to this, and as well PAMPs, will lead to the stimulation of these dendritic cells to mature. Soon after that, these mature antigen expressing dendritic cells, presenting dendritic cells, take residence in lymph node to begin the adaptive immune response. So as we said, this figure describes the sequence of innate immune response events occur when, as a result of a cut or an invader like bacteria got internalized. So let's talk about the second subtopic, which is the cell adhesion molecules. So immune cells interact actually with surface molecules or vascular epithelia to direct movement and to recruit cells to the sites of trauma. That's what we have just seen in the last figure. So we have two systems actually that provide this cell recognition, either selectins or integrins. So selectins are smallest family. These are the smallest family of adhesion molecules. They belong to a family called C-lectin family, and C came from the calcium ion, which is requirement for the binding with the leukocytes. So selectins can initiate cell-to-cell contact. And actually this, although it's a weak interaction, but it must overcome shearing force of the bloodstream, because as you know, the rolling effect of these selectins when bound to the leukocytes need to overcome the shear force from the bloodstream, although as well it is a weak interaction. So for selectins, we have a family of three carbohydrate binding proteins, either L-selectin, E-selectin, or P-selectin. So selectins generally share the all the three types of selectin share a common structure of 5 different domains. So actually, domain is a region of in a protein that have a specific function or a specific role in that protein. So selectins share a common structure of 5 different domains. As you will see, we have the N-terminal domain, which actually the carbohydrate recognition domain, CRD, which actually is responsible for the binding with the leukocyte. As well, we have the epidermal growth factor-like domain, and we have varying number of short consensus repeats, and you can see it's a varying number because actually this one, which is differentiating between the L, the E, and the P selectin, the length of this third domain. And then you have the fourth domain, which is transmembrane, and obviously from its name, because it's transversing the membrane, the transmembrane region. And you have the final domain, which is the only domain that is cytoplasmic domain, and actually it is the C-terminal domain of the protein. So these are the 5 domains that are shared between the 3 types of selectins. You need to understand that the CRD and EGF domains are highly homologous between the three selectins, so they are actually the same. While the other 3 domains, the transmembrane and cytoplasmic and so on, are not conserved across selectins. And this will show some structural diversity between across the non-conservation shows a structural diversity of selectins. So let's talk about these more details about the 3 types of the selectins. The L-selectin, which is actually sometimes is called CD62L, is found on most leukocytes, and they are necessary for lymphocyte entry into the immune organs, and they direct a secondary tethering of neutrophils, as well as its binding partners are the E-selectin and the cutaneous lymphocyte antigen, CLA. While the E-selectin, which is termed sometimes CD62E, it's found only on endothelial cells. And the expression of this type of selectin is induced by interleukin-1 beta, TNF-alpha, TNF-beta, and the lipopolysaccharides. The expression as well of this E-selectin on the gene level is induced by the NF-kappa beta, which is a pro-inflammatory transcription factor. It's a downregulation. That means its job is done. It's a downregulation, and it's a breakage. In other words, it occurred by its internalization from the surface of the endothelial cells and routed to the lysosomes for the recyclation of its amino acids. For the P-selectin, which is sometimes as well called CD62P, it's found as well on endothelial and the platelets. And actually, these kind of selectin are pre-packaged in these Webbel bodies, and they are rapidly expressed on the surface upon the induction of by histamine, either histamine, as you can see from the figure, the red spots on the surface of the membrane, once induction occur by histamine or thrombin or even oxygen radicals. They can be downregulated when the job is done as well by internalization, and can be recycled from endosomes to again to the secretory granules. As well, it can be induced by TNF alpha and the liposaccharides. So let's talk now about integrins. So integrins, actually, it's a chemokines as well, arise from the inflammatory site and induce a tighter contact to stop leukocyte rolling. And if you remember from actually the first or the second lecture, we have seen integrin, which actually the last complex that binds to the neutrophil, to the neutrophils or the leukocyte before extravasation. So actually, the chemokines naturally are immobilized to protoglycans. The expression of these integrins on the surface of the endothelial cells is activated by the binding of two important factors. The binding of the platelet activating factor from the endothelial cells with its receptor on the leukocyte. So that's why there is a binding during the process of rolling. And this, the activation of these types of chemokines can be induced by its own allosteric effects. So what about the pattern recognition receptor, PRR? Actually, it's an invariant recognition mechanism that detects specific unique molecular patterns associated with the pathogens and inflammation. And these are ancient, these PRR are ancient and genetically imprinted. These recognition molecules target molecular patterns that are also invariant and necessary for the pathogen survival. So if there is a pathogen that gain entry and needs a specific molecular proteins or molecular molecules and need for its survival, these pattern recognition receptors as well can recognize this pathway that is important for the pathogen survival and can attack these kind of pathways. And as well, it can target signal that coming from a pathogens like PAMPs and DAMPs as well. So these PRR are cellular bound or soluble and provide a surveillance mechanism, surveillance information to early responders for inflammation, like the mast cells, neutrophils, macrophage, and as well dendritic cells and B cells. There are two major receptor types screened for PAMPs or DAMPs. There are the toll-like receptors, which are actually found on the professional antigen-presenting cells, like the macrophage, dendritic cells, and B cells. And as well, there are other PRR, like the intracellular pathogens can be detected by something called NPSLRR proteins, which actually the nucleotide binding site and leucine-rich repeat, okay? So for these, for more understanding, these are the list of some selected pattern recognition receptor. So you can see toll-like receptor 1, 2, 3, 4. These are types of PR. There are more like more than these types. We'll see in the next slide. You can see from this table as well as the subcellular location, they are expressed because, as we said, it is either bound or soluble or these receptors. So you can see for most of these four types, the subcellular location is on the cell surface, and the ligand which activate these type of receptor from TLN1 and to 4, actually it differs, and these ligands actually can be considered as the PAMPs or DAMPs if they appear from the pathogen or the invader. And as well, in the fourth column, you can see the cell type, the different cell type that is expressed these kinds of toll-like receptor. You can see that dendritic cells might be unique cell for expressing toll-like receptor 3. Other types of toll-like receptor, you have a lot of other types. You can see toll-like receptor 7 as well. It's an intracellular compartment. It exists in intracellular, and the ligand for activation of this toll receptor actually it's a single-stranded RNA, and as well you can see the cell types that have these kind of receptor. For both of toll-like receptor 7 and 8, they can respond to a ligand which is a single-stranded RNA, and as well you can see the TR receptor 12, actually, it is still inside the intracellular component, but actually we didn't discover up till now what is its ligand responsible for the activation of its signaling pathway. The C-lectin receptor, considered one of the as well of the PRR, and it's on the cell surface as well, and it is activated by mannose and galactose, and exists in macrophage and dendritic cells. So for the fourth subtopic, we'll talk first on the neutrophils. So here we'll have all the info that is related to the neutrophils. Actually, they drive active killing of invading bacteria by either phagocytosis or even cytolysis. It is the most abundant white blood cells, represent almost 60% of white blood cells. They are multi-lobe nucleus, and they got the name because they are neutral to staining, actually. These are the first responders to inflammatory site, and cells which actually occur in circulation have a very short lifespan, hours to days, and can exist for the average is 6 hours in circulation. They have a longer lifespan in spleen, while the neutrophils are recruited to injury sites by process of chemotaxis through the compounds which is CCL8, which is interleukin 8, and as well as C5a and LTB4. They can occupy a temporary niche at the surface at the vascular lumen. They can be tethered by a transient interaction between the CD62L on the neutrophils and CD34 on the vascular wall, which causes the process of rolling over along the surface and the process of margination. They can be released in the presence of glucocorticoids and causing a temporary spike in the circulating neutrophils. And as well, they can release is driven by surface type of protease, which is surface metalloproteases, which is the ADAM8. The principal function of the neutrophils is phagocytosis, and that the target material is labeled. It needs to be labeled by C3b, Ig or IgG or collectins, type of proteins. And the process is triggered by the anaphylotoxins, the C3a, the C4a, and C5a. If you remember, this is actually the process of the opsonization, the opsonic phagocytosis. This is actually the process, opsonic and non-opsonic phagocytosis. So as well, neutrophils can kill internalized bacteria using the antibacterial peptides, even digestion by hydrolytic enzymes, and by oxidation by free radicals. Generation of free radicals can lead to the destruction of the internalized bacteria. The neutrophils have 3 types of secretory granules: azophilic granule, specifics, and as well gelatinase granules. All of these are packaged in special vesicles inside the neutrophils. Even the neutrophils can die by apoptosis to release its degraded material, and as well the accumulated debris, which constitutes the pus, the greenish color, This comes from a special type of enzyme inside the granules of the neutrophils, which is the myeloperoxidase. So here in this figure, you can see the process of phagocytosis by the neutrophils, or the process of clearance of the pathogen. So you can see here, there is a complement fixation through the, if you remember the process of opsonization, so we have the C3A, the C3B, and the MAC complexes are positioned over the bacteria. This will lead to the process of phagocytosis when binding of these components on the surface of the bacteria with its corresponding receptor on the surface of the neutrophils. And hence, the bacteria is opsonized and got phagocytosis by neutrophils. You can see as well that there is a lot of secretory granules inside the neutrophils that fuse with the phagosome. So you can see you have the azurophilic granule, you have the specific granules, and you have the gelatinase granules, different colors, roads, triangles, and all of them start to fuse with the phagosome. These granules have a lot of different types of enzymes, like the NADPH oxidase, the myeloperoxidase are activated. You can see the NADPH oxidase are activated. This will lead to the granules will deliver these antibacterial substances like defensin and lysosomes inside the phagosomes, leading eventually to killing the bacteria. And by the end of that, the neutrophil itself got exhausted and died. So these are the steps actually in the cytolysis by the oxidative burst that is provided by the neutrophil and all the enzymes that inside the neutrophils. The NADPH oxidase actually uses the oxygen to have free radicals, the oxygen-free radicals. And if you remember, the oxygen-free radical is one of the warfare that is used to destroy bacteria. And as well, myeloperoxidase uses as well hydrogen peroxide with a chloride ion to produce another free radical. While the catalase using hydrogen peroxide as well can break down hydrogen peroxide, and the superoxide SOD, superoxide dismutase, uses metal to combat these oxygen radicals. So, because as you know, the increased level of these radicals may affect our own healthy cells. So SOD is an internal antioxidant machine, one of the internal antioxidant machinery to decrease or equalize or regulate the amount of the free radicals inside our body. As well, the glutathione peroxidase as well can break down hydrogen peroxide, which is another precursor of free radicals, and can use this hydrogen peroxide, break it to form this water molecule, which is much safer than the hydrogen peroxide. So these actually, the protective measurements, the last two actually, last three actually, protective measures as an antioxidant mechanism, which can be provided actually with vitamin that we already eat. So basically here, you have your body using the reactive oxygen species or oxygen radicals as a type of the warfare that uses these radicals to attack bacteria. But in the same time, these radicals are very harmful to our own body. So we have an antioxidant mechanism inside our body to try to eliminate these free radicals, like the SOD and the glutathione peroxidase. But as well, we need vitamins as well, because a lot of vitamins is antioxidants. So what about basophils? So basophils, actually, it's an innate effector cell, have a multi-lobe nucleus and stains a dark purple blue color to the presence of a basic dye. Okay, that's why it's called a basophil, because it stains for a basic dye which is methylene blue. 1%, it is 1% of circulating leukocytes, short-lived in circulation, and they have a temporary residence in lymph nodes. The direct T helper cell differentiation, as we will see in the coming lectures, and as well play an defensive, active defensive role toward allergen with protease activity. They can release histamine, leukotrienes, and prostaglandins, and are a source for heparin and the BAF, platelet activating factor, as well as interleukin-4. The basophil express the FC receptor for the one of the antibodies, which is IgE, and as well can express MHC class 2 for antigen presenting for T helper 2 cells. They complete their development in the bone marrow prior to enter the circulation, and these basophils can enhance the humoral memory. For the eosinophils, these are innate effector cells that stain red, that's why eosino, with acidic dye eosine. They have a multi-lobed nucleus as well and numerous secretory vesicles. The interleukin-5 induced the bone marrow to release eosinophils, and it represented 2 to 5% of the circulating leukocytes. They are mediators of allergic inflammation. The secretory vesicles inside the eosinophils contain destructive proteins like acid phosphatase and eosinophil peroxidase, as well RNase, DNase, lipase, and plasminogen. They are capable of phagocytosis or degranulation. They can degranulation to release these specific effector proteins. They can perform cytolysis to flood the inflammatory site with high level of effector proteins. They can express FC receptor for another type of antibody, which is IgG, and MHC class 2, giving them the ability to activate CD4 cells, T helper cells as well. Eosinophils can secrete leukotrienes and prostaglandins. One of the major functions of eosinophils are defense to large extracellular parasites. They direct cell-mediated cellular cytotoxicity, CMCC. They deliver by delivering toxic free radicals and proteases to the parasite. Sometimes there are collateral host tissue damage, often results from the release of these cytotoxic agents because it might affect the nearby host normal tissue. The fourth type of the leukocyte is mast cells. And actually these are tissue-resident myeloid cells and do not ordinarily circulate, normally do not circulate. They are long-lived, found throughout the body, including rain, enriched at boundaries to environment where it can encounter pathogens are more likely. They may proliferate, differentiate, and mature inside the tissue, They can process trauma signals and can release injury-specific mediators, including histamine, serine proteases, and serotonin. They perform similar function to macrophage, including phagocytosis and antigen presenting. The principal function of mast cells is guarding from disturbance and to regulate inflammatory and adaptive immune response. Also function as sensors for trauma screening for DAMPs and PAMPs. They control the vascular integrity and initiate neutrophil recruitment. They express a specialized receptor for IgE called FC epsilon, a sensory mechanism actually for regenic molecules. It's a source for histamine, heparin, proteoglycan, and serine proteases. It can release reactive lipids such as thromboxane, prostaglandin D2, leukotrienes, and the platelet activating factor to permit neutrophil and macrophage intrusion. While the macrophage, which is a tissue-resident myeloid cells as well, recruited to the inflammatory site to complete the clearance of debris and wound healing. The monocyte, which actually can be considered as a precursor of the macrophage, monocyte in the circulation restore tissue resident macrophage, and this monocyte constitutes around 5% of circulating leukocytes. They can internalize bacteria, they kill it by using antibacterial peptides, and they can do digestion by hydrolytic enzymes and do exudation by free radicals. The expression of the microbial peptide, they can express microbial peptide using MHC class 2 and phospholipids by CD1. They can sequester bacteria by forming granulomas. The tissue resident of the macrophage are named according to the tissue. For liver, the macrophage is called Kupffer cell. For the dermis, histocytes. For the lung, alveolar macrophage. For the brain, microglia. For the bone, it's osteoclast. There are two functional states for macrophage, M1 and M2. M1 actually respond to bacteria and inhibit cancer growth, while M2 support angiogenesis, suppress immunity, and promote cancer growth. For the dendritic cells, the resident immature myeloid-derived dendritic cells are always collect samples from the interstitial fluid. It's like surveillance mechanism, and responsible for recognition of non-self antigens. While the mature dendritic cells, myeloid dendritic cells, take residence in secondary immune organs. The immature dendritic cells are in continuous for the process of processing antigen for presenting it to the T helper cells. The antigen is internalized by endocytosis and processed in lysosomes and inserted into and re-expressed back on the cell surface together with the MH class 2 molecule. The exposure to PAMPs induced the maturation of dendritic cells, and so it can migrate to lymph node. While the mature dendritic cells, it have a reduced capacity of endocytosis and increased the expression of MHC class 2. They increase the expression as well of co-receptor like CD80 and CD80-88 and can release interleukin 12, and these depend on the identity of the triggering PAMP. Here's this slide to remind you of the different immune organs. We already mentioned it in the first lecture. You have a primary and secondary. The primary are thymus and bone marrow, while the secondary are lymph node, spleen, and MALT. So let's see, after talking about the different types of white blood cells, we need to understand the process of hematopoiesis. So initially, during development, the site of the hematopoiesis change. So initially, during the fetus, early period of the fetus, the hematopoiesis occur in the yolk sac. This followed by increasing hematopoiesis in the liver and spleen. Soon after that, after birth, the distal long bone marrow take over the process of hematopoiesis until reaching adult. In the adults, the axial skeleton take the process of hematopoiesis. So you can see here, initially, the undifferentiated cells under the effect of interleukin-7, it can differentiate into lymphoid stem cells, while under the effect of interleukin-3 and CSF, it is differentiated into myeloid stem cells. And so it's clear now that the multipotent stem cells depend on the kind of the cytokine for the different differentiation stems of the lymphoid or the myeloid. The lymphoid can differentiate straight away to the NK cells. Under other condition of differentiation, it will give, the lymphoid will give the two progenitor for T and B cells, while under different activation condition and activation cofactors, it will give rise to the progenitor of other cell types. Soon after that, the precursor of T cells will soon differentiate in the thymus to thymocytes, and as well the progenitor for B cells will differentiate into B lymphocytes. While the other, in the myeloid stem cells, you can see that depending on the kind of the cytokines, it will give rise to the corresponding type of the white blood cells or platelets or red blood cells. After that, you can see that even the thymocytes the thymocyte can be differentiated, further differentiation into helper T lymphocyte, cytotoxic, while the B cells can be differentiated into plasma cells. Here you can see that the progenitor of a monocyte or granulocyte can give rise to dendritic cells, or even come from the monocyte, which is the major precursor of the macrophage. And you can see as well that the basophil is the precursor, the progenitor of the mast cells. Here as well, you can see the different lymphoid cells, T cell, B cell, NK cells, plasma cell, the different, these under the, this actually real image under the electron microscope, the granulocytes, neutrophils, eosinophils, basophils, and you can see as well the tissue-resident cells like the macrophage and the dendritic cells. Generally, the neutrophils represent mostly the highest percentage in the blood of the adult, while band cells represent up till to 5%, lymphocyte from 20 to 40%, monocyte up till 10%, eosinophils up till 5%, and basophil less than 1%. Band cells actually represent immature neutrophils. Here we can summarize the white blood cells. Initially, the myeloid cell, the neutrophils, is the most abundant circulating. It is granulocytes, and its description, lobular nuclei, 3 or 5 lobes, always looks like a small pink cytoplasmic granules in the cytoplasm. Its main function is phagocytic activity, killing extracellular pathogen. While the lymphoid cells, there is the lymphocytes. It's a secondary, actually, in the bloodstream, and can exist in the secondary lymphoid tissue. And its physical appearance actually looks large, owing to the large nucleus, and have very thin rim of the cytoplasm. The surface markers include, you have different types of lymphocyte. You have, depending on the type of the lymphocyte, you have its own specific marker. And the function, still at this stage, it have no function until activated to be differentiated to either B cell, T cell, helper, cytotoxic T cells. For the plasma cell, it is in the bloodstream as well as the secondary lymphoid tissue and bone marrow. It has small eccentric nucleus, stained, darkly stained, Golgi apparatus. It is actually the terminal differentiated form of the B cells that secrete antibodies. While the natural killer cells, it's only in the bloodstream, and it have large cytoplasmic granules, and its surface marker are CD16 and 56, and its main function, killing actually infected cells and cancer cells. Another type of myeloid cells is monocytes, which it's actually circulating cells, and it have no granules at all, and the nucleus looks like a kidney shape, and actually it is a precursor of tissue macrophage. While the macrophage itself, it is resident in all tissues, and it is a granulocyte with a cytoplasm, that a ruffled cytoplasm, a lot of cytoplasmic vacuoles and vesicles, phagocytosis. It can present antigen, and it is a T cell activator as well. For the dendritic cells, it can resident in epithelial and lymphoid tissue. It is a granulocyte as well, with thin stellate cytoplasmic projections, and as well it shares its function with the macrophage, actually doing the same function, phagocytosis, antigen presenting cells, which is the major function of it, and as well T cell activator. For the eosinophil, it is circulating as well, and actually one of the cells that is recruiting into the loose connective tissue of the respiratory and GI tract. It is granulocytes, bilobe nucleus, very large bilobe nucleus, majorly concerned with the elimination of extracellular parasites and the type 1 hypersensitivity. For the mast cell, it resides in most of our tissues adjacent to the blood vessel. It have a lot of granules, equipped with a lot of granules, have a large blue cytoplasm due to the granules, and as well it deals with the elimination of extracellular parasite and as well activate type 1 hypersensitivity. For the basophil, it low frequency in circulating blood, and it have a lot of granules as well, large bilobe nucleus, and as well it have the same function as a mast cell and the eosinophil. Thank you, and this conclude the lecture of the innate immune response.